Hello friends, in this video we'll be discussing about sickle cell anemia. It's also called as sickle cell disease. It's a blood disorder where red blood cells acquire sickle cell-like shapes when they are deprived of oxygen. When we see this disease at a molecular level, we see actually it's a point mutation where different kind of amino acid is produced at RNA level. The sickle cell disease is also called anemia because it's the decrease in the total amount of blood or hemoglobin. Here in this diagram, you can see the red blood cell transforms its shape into sickle cell-like, so-called sickle cell anemia also. Now let's see the genetics of it. The HBB gene, which is responsible for sickle cell anemia, is present on chromosome number 11 on its short arm, which is at the position of 15.5. It has a beta globin locus on it, which produces HBB protein. Here in this diagram, you can see the position of HBB gene. So, what the sickle cell disease is, how this red blood cell acquires sickle cell-like shape, it's all due to defective hemoglobin, which gets mutated at DNA level. We have the normal structure of hemoglobin like this. It has two alpha chains and two beta chains, and it's the beta globin that gets misshapen, with which this red blood cells at the time of lack of oxygen, deprived of oxygen, it gets misshaped. And this crystalline structure, the abnormal structure of hemoglobin, is due to the point mutation when we see this at DNA level. It's the error in the DNA replication. Now we will see the cause behind mutation. First of all, we draw here the normal hemoglobin, that's HbN. It has got polypeptide chain. 5th, 6th, 7th amino acid of polypeptide chain is proline, glutamic acid, and 7th is also a glutamic acid. But in case of defective hemoglobin, this glutamic acid is replaced by valine. So here the 5th amino acid will be proline and 6th amino acid will be valine instead of glutamic acid in defective hemoglobin. And this valine has a property to repel water, that's it's hydrophobic in nature, instead of glutamic acid, which was hydrophilic in nature. And this makes the two different entities of hemoglobin, because the first one, it has a glutamic acid which is hydrophilic in nature, and another one, that's the defective hemoglobin, it's valine, it has got hydrophobic amino acid in it. When we see this mutation at molecular level, we have two different cells here, that's normal cell which has got normal hemoglobin, and sickle cell which has got defective hemoglobin in it. And here we have the three levels. First one is DNA replication, second one is transcription, that's formation of RNA, and third one is translation, where proteins are formed from amino acids. When we see this process of replication in normal red blood cell, we have GAG sequence, that's guanine, adenine, and guanine, which got complement sequences cytosine, thymine, and cytosine, which forms RNA strand as GAG that codes for an amino acid called glutamic acid. On the other hand, we have sickle cell which has got defective HBB gene, and this defective gene is only and only due to the replacement of adenine by thymine base in a DNA strand. And in its complement strand we get CAC bases, and when there is formation of RNA strand we will get it as guanine, uracil, and guanine, because in RNA strand we have uracil in place of thymine. And this genetic code, that's GUG, codes for valine amino acid in sickle cell. Here you can see the HBS, that's defective hemoglobin, which has got mutated HBB gene. It transports O2 very well. Under normal conditions it transports O2 very well, but it's only when these hemoglobin molecule releases O2 in tissues, and this hemoglobin gets deformed into crystalline structure, and this crystalline structure is called a tactoid. And then these tactoids form sticky ends, and with the help of hydrophobic ends of beta globin chain, they interact with each other and clump, and this formation of clumping is called sickling. After the process of sickling, these defective cells adhere to the endothelium of blood vessel, where they cause vaso-occlusion, and after that there is a rupture of cell membrane, and this causes the total anemia. That's why we call it sickle cell anemia. Guys, thanks for watching the video. Do share this video and do not forget to subscribe this channel.